Pinnacle says, life is just the way the proton body exists. Through existence, human beings give life more profound meaning by active proteasome enzyme. Vazyme deciphers the mystery of vitality, comprehend the depth and significance of life issues. Vazyme is using cutting-edge technology, contribute to the rise of national life sciences. After years of unremitting exploration, Vazyme has achieved key breakthroughs on the two technology platforms of enzyme engineering and antibody engineering. Superb sufficient proton and antibody production and purification technology is the application of performance and quality level of the product, thus widely recognized by customers in the industry. Vazyme has superb enzyme-directed evolution mutation technology and efficient recombinant expression technology that improve the performance of the enzyme. And through the scale effect, the production cost was significantly reduced based on the strong R&D strength oriented by real-time market demand. Vazyme is always ready to respond to genetic engineering, provide better quality enzymes, mutants, and antibodies, provide customers with diversified innovative products and solutions. The company focuses on providing overall solutions, products, and services in the field of biomedicine, aimed at early warning of serious illness through preventive treatment, formed a development pattern with in vitro diagnostic products as the main product, enzyme and antibody raw materials products as the characteristics. Products cover in vitro diagnostics, high-throughput sequencing, life science research, and other fields. In vitro diagnostics focuses on the research and development, production, sales, and service of products and instruments. Products cover cardio-cerebrovascular, prenatal and postnatal care, kidney function, infection, children's respiratory tract, chronic disease management, and other fields. Vazyme tirelessly polishes and sculpts its products with almost engraved craftsmanship. Committed to innovation, committed to providing products and services with excellent performance for the field of biomedicine and life science research. During the novel coronavirus epidemic, Vazyme actively organized research and development quickly, launched novel coronavirus detection series kits, and obtained certification from many overseas regions and countries, contribute to global epidemic prevention and control. Vazyme insists on creating value for customers, focus on this driver, provide employees with a broad development platform, attract the best talents from the country and the world to join. With the concept of making outstanding contributions to the progress of human health, take on the mission of promoting the development of China's life science industry. Keep moving forward.
In a few large antivirus battles in human history, when virus persecuted us silently, we didn't surrender. However, to defeat the invisible killer in human body, we need to know it. Only by knowing it as soon as possible can we obtain more time to conquer it, prevent more misfortunes from happening. The progress of human science shortens the time to discover virus. From over 5 months to discover SARS in 2003, to over 1 month for H7N9 in 2013, even now to only 5 days for the whole genome sequencing of SARS-CoV-2 with NGS technology. The second-generation NGS technology, mNGS, is used for SARS-CoV-2 sequencing. Isolation, cultivation, and purification of microorganisms are no longer needed. Nucleic acid could be extracted directly from clinical samples to build the virus library and get genetic information of pathogen from samples via NGS. Then, virus sequence, the picture of the virus, can be attained with bioinformatic analyses. The picture will be helpful to capture the virus. 3 ways to capture the virus are nucleic acid detection, antibody detection, and antigen detection. An all-round approach, leaving no place for virus to hide. The most rigorous and accurate method for nucleic acid detection is to extract nucleic acid from samples of secretion in respiratory tract, take the conserved sequences in viral genomes as targets, and design the corresponding primer and probe. According to the real-time quantitative fluorescence PCR technology, the target sequence index increases, and fluorescent signals appear after each round of amplification, during which fluorescent signals are accumulated and detected by relevant PCR instruments, while samples not infected by the virus will not show any PCR reaction due to the absence of relevant target sequence. Whether the sample contains the virus can be judged through fluorescent signals. The other 2 ways for detection are antibody and antigen detection. After initial infection of COVID-19, B cells are primarily activated to generate specific antibodies against the virus. Antibody detection demonstrates health conditions of patients through the detection of their specific antibodies. IgM, first antibody generated, remains in early and middle stage of disease onset and cannot be detected after body recovered for a period of time. IgG can be detected about a week after disease onset and used for diagnosis and tracing back infection history. The antigen detection targets at COVID-19 viral antigens. Its genes encodes multiple structural proteins, like N protein and S protein, with several antigenic epitopes. Because of binding between specific antigen and antibody, we can identify the existence of SARS-CoV-2 antigens, directly demonstrating if the sample contains antigens for COVID-19 virus. These methods in different methodologies vary in testing conditions, operational standards, and individual immunity. After knowing characteristics of the virus through detection, we take countermeasures. Our immune system identifies pathogens after viral infection, creates specific cellular and humoral immune protection against COVID-19 virus, to remove pathogens, and generates immune memory cells to prevent secondary infection. In vitro building of system for immune recognition, protection, and memory against the pathogen in recovered individuals by simulation is helpful for the development of immunotherapy drugs and vaccine. Specific protective antibodies or antiserums developed against the COVID virus are effective for treatment under different scenarios. Vaccine carrying protective epitope information of pathogens recognized by immune system can simulate the process that pathogens stimulate immune system for specific protective immune responses and for immune memory. Attacked by the virus again, immune memory cells with protective epitopes will be rapidly activated to protect body and prevent viral intrusion and replication. All researchers are working against time for vaccine development. Evaluations on safety and efficacy are the core in clinical trials of vaccine. In Phase 1 and 2 clinical trials, the focus is on the dosage and procedure for immunization. Except monitoring and adverse reactions for vaccination, we should detect humoral immunity and cellular immunity to assess the immunogenicity of vaccine, indirectly reflect its efficacy. Large-scale Phase 3 clinical trials will be conducted in badly hit areas. Vaccine efficacy will be evaluated by comparing infection rate of vaccine group and placebo group. As time for R&D of COVID vaccine is greatly shortened, vaccine efficacy and persistence should be tested continuously after its launching. Since the formation of the universe, there have been countless battles between humans and virus. With strong desire for survival and life extension, virus constantly evolved and mutated. In such ceaseless war, human technology is advancing simultaneously, accelerating technological innovation, and achieving significant technological process in life safety and biosafety. Developing technologies to improve human health